She was born in complete silence. Her parents learned the diagnosis when she was just a few months old. Profound deafness, bilateral, both ears. The gene responsible for transmitting sound signals from her inner ear to her brain had a mutation that made it non-functional. The protein her cochlea needed to convert vibrations into electrical signals simply did not exist in her body. Her parents were told what parents in that situation have been told for generations. She would need hearing aids, then cochlear implants. She would learn sign language. She would adapt. There was no treatment that could fix what was broken at the genetic level. That answer had been medically accurate for the entire history of modern medicine. Then a team at Fudan University's eye and ENT hospital in Shanghai decided to change it. They injected a working copy of the broken gene directly into her inner ear through a single needle. One injection, no surgery, no implant, no device attached to her skull. Within 3 weeks, she began responding to sounds for the first time in her life. Within months, she was imitating words, then short sentences, then reciting poems, then singing. Her parents, who had spent years learning how to communicate without sound, suddenly had to remember to be quiet at bedtime because their daughter could now hear them talking in the next room. To understand why what happened in China is so extraordinary, you need to understand what hereditary deafness actually is, and why medicine spent a century unable to touch it. The human ear converts sound into electrical signals through a structure called the cochlea, a spiral-shaped organ in the inner ear lined with thousands of specialized hair cells. These hair cells detect vibrations at different frequencies and convert them into signals that the auditory nerve carries to the brain. For this conversion to work, the hair cells need a protein called otoferlin. Otoferlin acts as the messenger at the junction between the hair cell and the nerve. When sound arrives, otoferlin triggers the release of the chemical signals that tell the auditory nerve to fire. Without otoferlin, the hair cells can detect sound perfectly. The cochlea is structurally intact. The auditory nerve is intact, but the signal never crosses the gap. The brain hears nothing. This condition, caused by mutations in the gene OTOF, is one of the most common forms of hereditary deafness. It affects children born with completely intact ears that simply cannot communicate with their own brains. For over 100 years, the only options were mechanical. Hearing aids amplify sound, but they cannot help if the conversion mechanism is broken. Cochlear implants bypass the hair cells entirely and stimulate the auditory nerve directly with electrodes surgically inserted into the cochlea. They are remarkable devices that have given millions of deaf people access to sound, but they are also permanent implants that require surgery, that carry risks, that last only so long, that produce a version of sound that takes years of therapy to learn to interpret, and they still rely on a functioning auditory nerve. They do not fix anything. They route around the problem. The gene that causes the problem stays broken. The protein that is missing stays missing. What Chinese researchers at Fudan University, in collaboration with Zhongbao Hospital at Southeast University, and several other institutions across China, asked was a different question entirely. What if you could simply deliver the working gene to the cells that need it? Gene therapy is not a new idea. Scientists have understood for decades that if you could deliver a functional copy of a broken gene into the cells where the gene is supposed to work, you could potentially restore that function. The challenge has always been delivery. Genes are large molecules. Cells do not simply absorb genetic material from the outside. You need a vehicle, something capable of entering the cell and depositing its genetic cargo without triggering an immune response or causing damage. The vehicle researchers chose is called an adeno-associated virus, or AV. It is a naturally occurring virus that has been stripped of the parts that make it harmful and reprogrammed to carry a therapeutic gene instead of its own genetic material. The problem with the OTOF gene specifically is that it is unusually large, too large to fit inside a standard AV. The Chinese research team, in collaboration with partners including Massachusetts Eye and Ear and Harvard Medical School, solved this by splitting the gene across 2 separate viral vectors that recombine inside the cell with remarkable efficiency, around 90% of the time, producing a functional copy of the protein. The solution was elegant, technically demanding, and years in the making. The first child in the world to receive this treatment was treated in December 2022 at Fudan University in Shanghai. The results were rapid enough to surprise the researchers themselves. Within weeks, the child began responding to sounds. The trial expanded. By 2024, 5 children had received treatment in both ears simultaneously, an approach that produced even stronger results. By 2025, The trial had grown to 42 participants across 8 clinical sites in China, including children as young as 9 months and adults as old as 18. The results published in Nature in April 2026 were striking. 90% of participants experienced significant and lasting hearing improvement. Before treatment, participants could not detect sounds below 106 decibels. After treatment, the average threshold dropped to 52 decibels, the range of normal conversation. Of the 15 participants followed for 2 years, all could hear conversational speech. 60% could hear whispers. One 11-year-old girl, who had been born profoundly deaf and had never used a cochlear implant, began speaking simple words after treatment. Professor Eli Xu, who led the clinical work at Fudan University and co-authored the Nature paper, described what he saw in his patients over time. They progressed from responding to sounds, to imitating speech, to speaking in short sentences, then to reciting poems, and even singing. They always fill us with joy and encouragement. What makes the Chinese trials particularly significant is not just the results, but the scale and speed at which they were conducted. While researchers in the United States and Europe were running small pilot studies, China built a network of 8 clinical sites, enrolled 42 patients, followed them for up to 2 years, and published comprehensive long-term data in Nature and Nature Medicine. The Chinese government had identified gene therapy as a national research priority. Funding flowed. Institutions collaborated. The regulatory environment in China allowed the trials to move faster than the American system permitted. In the United States, the first children did not receive the treatment until 2023, a full year after China. As of the time of writing, American trials are still in early stages. The longest follow-up data, the most comprehensive safety record, and the largest patient cohort all come from China. Now here is what this means for the roughly 26 million Americans living with significant hearing loss, and for the estimated 1.5 in 1,000 children born with congenital deafness every year. The OTOF mutation, which is what these trials target, accounts for only about 2 to 8% of hereditary deafness cases. It is not the most common cause, but the researchers who led these trials have said publicly and clearly that OTOF is just the beginning. The same gene therapy platform, the same AV delivery system, the same infrastructure of clinical sites is now being adapted to target other genes, including GJB2 and TMC1, which together account for a much larger share of hereditary deafness worldwide. Animal studies on these other mutations are already returning promising results. The researchers are confident that what worked for OTOF can be extended systematically and iteratively to other forms of genetic deafness. The platform is proven. The safety record is clean. No serious adverse events have been reported across dozens of patients and years of follow-up. What took a century to begin is now accelerating rapidly, and the acceleration is happening primarily in China. The FDA approved the first gene therapy for OTOF deafness in the United States in May 2026, developed by Regeneron. That is a meaningful step. But the therapy Americans will receive was not built from scratch in American laboratories. The foundational clinical evidence that made regulatory approval possible, the proof that this approach works in humans, was generated in trials conducted in China, at Fudan University, at Southeast University, at hospitals across Shanghai and Nanjing and Beijing. American regulators looked at Chinese clinical data and used it to approve an American product. The country that proved the concept was China. The country that built the largest trial, followed the most patients, and published the most comprehensive long-term data was China. And the researchers who described watching a child who had never heard a sound in her life begin to sing, for the first time, were working in Shanghai.